We welcome you all for the most awaited distinguished lectures by the Nobel Laureate, Sir Professor Richard Roberts. I would like to call upon dais our Vice Chancellor, Professor B.J. Rao, to introduce the Nobel Laureate, Professor Richard J. Roberts. And also I would welcome Sir Professor Richard Roberts onto the dais.
Good afternoon, all of you. I think you should find some chairs. There are chairs. All of you. Please be seated. It's going to be a very interesting talk. Oh, there is a live relay in seminar hall. Instead of standing here, you can move to seminar hall also, where the live relay of the lecture will happen, is happening. All of you, please settle down. Or move to the next lecture room. Thank you. Thank you very much. I want to warmly welcome Professor Richard Roberts to this campus, to Hyderabad city, and to India. Richard Roberts has been a frequent visitor to India. He knows us. I believe he did visit University of Hyderabad some 10-12 years ago. So we are welcoming him again to the University of Hyderabad. Please welcome with an applause. I will briefly give his profile. He is a biochemist by training. He received his education in the University of Sheffield, where he earned his Bachelor of Science degree in chemistry. Later, he did his PhD in organic chemistry and did his postdoctoral work at Harvard University. He is best known for his groundbreaking work in the field of genetics, particularly for his contributions to the discovery of split genes, mRNA splicing. Dr. Roberts, along with Phil Sharp, were awarded the Nobel Prize in Physiology and Medicine in 1993 for their discovery of split genes and RNA splicing, which actually revolutionized our understanding of gene structure, function in all organisms, all eukaryotic organisms. Throughout his career, Roberts has held various research positions at institutions such as Cold Spring Harbor, New England Biolabs, etc., where he has made significant fundamental contributions to the understanding of DNA, RNA, structure, function in the context of biological regulations, and significantly contributed to our understanding of genetic diseases. Throughout his career, Dr. Roberts has held various research positions at important organizations such as Cold Spring Harbor, New England Biolabs, etc. In addition to his scientific research, Dr. Roberts is also a serious advocate for open access journals to scientific information dissemination. He has been involved in various efforts to promote transparency and accessibility in scientific publishing enterprise. Dr. Roberts has received numerous honors and very significant honors. I will only list a few. Nobel Prize in Physiology or Medicine 1993, Albert Lasker Award for Basic Medical Sciences in 1991, Louisa Gross Howich Prize 1989, Gairdner Foundation International Award 1986, Sir Hans Krebs Medal 1992, National Medal of Science 1992, Golden Plate Award 1994, Fellow of Royal Society, London 1994, and our own foreign member in the National Academy of Sciences 1986. He has had very significant contributions in the area of splicing genes, in the area of restriction modification enzymes. In fact, he has about 70% of restriction enzyme class 2 to his own contribution. It's our privilege and pleasure to invite him to University of Hyderabad on the auspicious occasion of BioAnveshana-2024, on the occasion of University of Hyderabad celebrating its Golden Jubilee year. Thank you very much. Please applaud Dr. Roberts. Thank you very much. Thank you. This is your microphone.
Okay. Okay, thank you. Okay, well, it's a pleasure to be here. I always enjoy coming to India. I found tremendous hospitality, but I find that wherever I go, where there are scientists, and you will discover the same, you have a friend. One of the great things about science is it brings everybody together. We can disagree vehemently about things, but we don't find it necessary to shoot one another. I think if politicians learnt a little about science, they might do a lot better in this world. Anyway, what I'm going to do today is to give you a very personal history of my own life and how I came to win the Nobel Prize. And along the way, I'm going to try and give you some ideas of things that you might want to look into in your own lives as you go through science. And one of the overlying things is luck. I think we are all lucky. Everybody has a lucky break. If you look back on your life and you say, well, you know, I went this way rather than this way because of luck. And it is very important. It was a lesson that I learned when I was 16, 17 years old. I'll tell you a little more about it later. But it really is very important. Luck plays a huge role in our lives. And the secret is to take advantage of it, not to pretend that you somehow shouldn't have been lucky. Everybody is lucky. My luck actually started fairly early on. I was born in this town, Derby, in the middle of England, in 1943, 2 years before World War II ended. And it turned out the street that I was living on was on the flight path between Germany and the Rolls-Royce factory where they were making the engines for the bombers and fighters and so on that were attacking Germany. And they were constantly trying to bomb the Rolls-Royce factory. I lived on a street such that one side of the street was almost totally destroyed by errant bombing. I lived on the side that survived. So here I am, 2 years old, and already I had tremendous luck in my life just by living on the correct side of the street. It's surprising when you look back just how little things like that make a huge difference to you. And it's something you should all do. You should all take some time to go back and think about ways in which you were lucky. Now, when I was 4 years old, my father, who was a motor mechanic, moved down to Bath in the southwest of England, a beautiful city in Bath. And so I ended up growing up in Bath. I remember almost nothing about Derby. And that turned out to be very nice too. A lot of good things happened in Bath that may not have happened had I stayed in Derby. One of the first things that happened was I went to this junior school, St. Stephen's School, from 1950, from when I was 7 onwards. And it turned out, after I'd been there for a year or two, the headmaster, at the end of the day, used to come and give me puzzles to do. Give me a slip of paper, and he said, here's a puzzle. And they were pretty much all mathematical puzzles. And as a result of that, I fell in love with mathematics. I really felt, if you'd asked me when I was 7, 8, 9 years old, I was going to be a mathematician. This was what I was going to do with my life, and all because of this headmaster. And it turned out, I thought he did this to everybody, but he didn't. It was just to me. He somehow saw that, I could do something, I would be interested in this, and it made a big difference to me. And I'm still in love with mathematics, and what I do professionally is mathematics, as you will see later on. So that was really good. It was a very good experience. The other thing that happened that was interesting was I had the best exam experience I've ever had. It turned out one day we roll up at school and our class, we have to sit and got a few sheets of paper, and the teacher said, can you fill in the answers to these questions on the sheet of paper? It turned out it was something called the 11+ examination. And depending upon how well you did, you either went to grammar school, which was sort of headed towards a more intellectual future, or you went to a technical school, which was heading into a technical. I had no idea, none of us, we had no idea that this was an exam that was actually going to shape the future for us. And I think it's actually a very good way to do exams. This idea of swotting for exams, I did spend about a year doing that, and it made absolutely no difference to the score I got at the end of it. It was a waste of time. So I think we all need to think a little bit about what is the best way to set exams and whether or not you really need to study. If you've been paying attention in class, you will know everything that you need to know for the exam. So that was good. So by the time I was 11, I changed my view of what I wanted to do in life. I'd read a book. It was called How I Became a Detective, and about, how you normally become a police detective. And that sounded fantastic. And by the time I finished that book, I wanted to be a detective. I knew that this was what I was going to do with my life. And then, just after that, my father bought me a chemistry set for Christmas, and these were days where chemistry sets really had all the interesting chemicals in them. You buy one today, they're boring, they're dull, nothing interesting. But mine, I discovered I could make fireworks, I could make explosives, which were in the chemicals there. And so then I decided I wanted to be a chemist. So here we are, we've gone from mathematics to being a detective to being a chemist. And chemistry stuck with me because I loved the experimental side of chemistry. I like the fact that you go in and, do some experiments and everything works out well if you've done them well. So that was good. The pictures are just pictures of Bath. This is the Royal Crescent, a beautiful Georgian architecture. And these are actually the Roman baths. Bath was settled originally by locals in England, and then the Romans came along, discovered that the hot springs were there, and they put in these baths, which are really quite spectacular. Very good bath. Now, after you age 11, you go on to higher education. I went to the City of Bath Boys' School, a grammar school, and had a really good time there for the most part. And we had a very good math teacher, and so I got very much excited about doing mathematics. I also learned to play the violin while I was there, and I joined the school orchestra. Now, Bath is situated very close to this limestone escarpment called the Mendip Hills, and I was introduced to caving. We used to go into the caves and go exploring and collecting insects and doing all sorts of things, and I love that, absolutely love that. I still do, although I don't do it very often. But last year I was in Beirut and went to this cave in Beirut. It's a tourist cave, but it was fantastic. It's one of the best tourist caves I've ever been in. So I love that. That was good. And I also discovered jazz. I rather like jazz. I like classical music, but I like jazz also. Now this is where I first learned the importance of luck. So when I was 18, I got very bored at school and I pretty much stopped going because all they were doing was repeating what we'd learned the year before just for the purpose of taking an exam. And I used to go in the morning, sign in, and then I would leave and I'd go and play billiards and snooker at the local YMCA, and then at the end of the day I'd go and sign in to indicate that I'd been there all day. And there was one physics teacher, really nasty man. He was not nice at all. And he discovered that I was doing this, and he tried to get me thrown out of school. And I thought I was going to be thrown out. I really did not think that they were going to let me stay on. And the thing I did best at the time was to play billiards and snooker. And so I went to audition to become a professional billiards player with a guy who was then the world champion. But before I found out whether I could get the job as his sparring partner, he was going around giving exhibition lessons, the headmaster relented and said, well, we're not going to throw you out of school, but you really have to stay all day and we're going to monitor it and so on. And so I thought I missed the chance to be really famous. I could have been world snooker champion, I tell you. I was good. But I was playing one day, and I made a shot, and instead of potting the ball that I was trying to pot, I potted another one. And then I played the next shot, and I muffed it. I messed it up completely. And afterwards, an old gentleman came up to me, must have been 80, 85 years old. He came up and he said, you could be really good at this game, but you have to learn the key lesson is when you get a lucky break, you've got to concentrate twice as hard on the next shot to make sure you take advantage of it. And that's something that stayed with me throughout my life. Every time something lucky happens, I always think, well, how can I make the most of this? What can I do so that I don't waste the luck? And I think you should all think about this in your own lives. Don't feel guilty if you're lucky and your friend isn't, or somebody you know isn't. And so this, for me, was really an important life lesson. And at the end of this, I'll tell you how it's played out. When it came time to go to university, I had missed a year of school because of this year that I took off, because I failed physics the first time around. I had to retake the A-levels. Couldn't go to Oxford or Cambridge, although in retrospect that was probably good. And there were two places that had really good chemistry departments. One was Sheffield University, the other was Southampton University. I applied to both. Only Sheffield accepted me. Southampton didn't want me. But it was lucky. Sheffield worked out for me to be exactly the kind of place that I should have gone to, and I actually knew some people who went to Southampton and they didn't have as good a time as I did at Sheffield. So I stayed there, did chemistry, and it was really good. I did the bachelor's, then I did a PhD there. And something happened during this PhD that was really incredible. This gentleman, this Japanese man, Kazu Kurosawa, was a postdoc during my first year as a graduate student. And I learned more from him about how to do chemistry than from anybody I've ever learned anything from in my whole life. He was fantastic. He could teach you what you should do, why you were doing it, help you interpret the results. If you failed, he knew exactly why and could troubleshoot it. I learned so much from him that in my first year, I had enough results for my PhD. But in England, you have to wait 3 years. They're very fussy, these people. They make me stay there for 3 years. Kazu went back to Japan and got a nice job there, and I started in the second year doing experiments I thought might be good, but they didn't really work out so well. They were okay, but they were not great. But I spent a lot of time reading. I used to go over to the library and read all the time. And I read this book called The Thread of Life by John Kendrew. And this book changed my life again. This book, John Kendrew was a Nobel Prize winner. He won the prize for protein structures, one of the first people to do a protein structure. But it was really all about the origins of molecular biology, how it all got started. And I was just blown away. I thought this sounded so interesting. And so I decided I was going to switch and become a molecular biologist. And so when it came to time to apply for a postdoc, I would talk to, my professor who I work for, who said, well, there's no such subject as molecular biology. In those days, chemists didn't think that there was such a subject. There was one young assistant professor there who did think that it was okay, and so, but I went away anyway. I have to tell you, one of my problems, I'm a very poor listener. If people try to give me advice and tell me, oh, you should do this or you should do that, I usually think, well, that's going to be boring, because he thought about it or she thought about it. I want to do what I've thought about, not what somebody else tells me. So I tend always to go against the grain when I'm given advice. But this book was terrific, and so I applied for 6 postdoctoral positions in places where I could do molecular biology. Of those 6 places I applied to, only one man accepted me. And that was a man called Jack Strominger. And at the time, he was in Wisconsin. He was at Madison, Wisconsin. And 2 months before I was scheduled to go and join him, he wrote to me and he said, could you wait for 1 month because I've just been made a full professor at Harvard. And so, a lucky break, by chance, I ended up at Harvard instead of Wisconsin. And that turned out to be very good for a number of reasons, but again, a lucky break. And, if you think, well, this guy's pretty lucky, I've not told you anything yet. A lot more is coming. So I ended up being given a job to sequence a tRNA, and I was picking this up from a postdoc, an Australian postdoc who was leaving, and so he sort of taught me how to do what he was doing, but I didn't like the way he was doing it. And I also started to read the literature and heard about Fred Sanger and his new method for sequencing RNA. And so I wrote to Fred and I said, could I come and learn your methods? He was at the MRC lab in England and in Cambridge. And Jack said, well, I've got the money, I'll pay for you to go. So I went, I spent 6 weeks in Fred Sanger's lab. Fred is an interesting guy. One of the few people who have won 2 Nobel Prizes and probably should have won a 3rd. He developed all the methods for sequencing proteins. That was his first prize. He then developed the methods for sequencing RNA, but he never got a prize for that. He should have done. And then he developed methods for sequencing DNA, which turned out to be much better than the method that he shared the prize with, Wally Gilbert. His method actually set sequencing back, if anything, rather than bringing it forward. But for some reason, the Nobel Committee thought he should get it. Anyway, I spent 6 weeks in Fred's lab, learned how to do everything, came back and set up RNA sequencing at Harvard. And that turned out to be really, really good. For one reason, it meant that I sort of had to get everything started from scratch, and it's always good to set up a technology lab, if you like, all by yourself. But also what happened was that many people came to me to learn how to do it, because it turned out, by chance, I was the first person in the Boston area to be doing this. Everybody else were using older-fashioned methods at the time. So as a result of that, I met a man called Mark Potashny, who at the time was an assistant professor at Harvard. He eventually became full professor and then went down to Columbia. And one day Mark Potashny comes up to me and he says, Jim Watson Watson of double helix fame, he said, Watson is going to come and talk to you about a job at Cold Spring Harbor. And so 2 weeks went by, and then Mark came up to me and he said, well, Jim doesn't actually know who you are. Maybe you could go and introduce yourself. And so I did. I go knock on his office. I was in his office for a maximum of 10 minutes. It may well have been less. As soon as I got in, he said, I want you to come and sequence SV40 DNA at Cold Spring Harbor. and told me this was a job. And as I was leaving the office, he said, you probably should come down to Cold Spring Harbor and see what it's all about. But you've got the job, he said, don't worry. I mean, it was not an interview, it was more a statement of fact that this was something he thought I should do. And I figured he must have talked to Strominger. Strominger knew nothing about it. And it was all Mark Potashny who had convinced Jim that he should give me the job. So anyway, the bottom line was I went to Cold Spring Harbor and intending to sequence SV40 DNA using the methods that Fred Sanger had developed by copying the DNA into RNA and then assembling the sequence. But as soon as I got to Cold Spring Harbor, I learned that there were 2 other groups already doing this. And I thought, I don't want to be the third, these other guys are doing it, and they've been doing it for some time. And I don't like competition. I really don't like to compete with. I want to do something that's different. And I was fortunate, I'd been to a lecture at Harvard by this man, Dan Nathans, up here. And he and Werner Arber and Ham Smith won the Nobel Prize for learning or discovering restriction enzymes and then learning how they could be used. And so Dan talked about the fact that with SV40 DNA and this enzyme endonuclease R that Ham Smith had discovered, you could cut SV40 DNA into a number of discrete pieces. And one of the reasons that RNA sequence was developed before DNA sequence is because you need small molecules to practice on. I'm sure you all know, anytime you think about doing something brand new, you've got to have something small to practice on. So if you've got a new idea for doing something, you want to know that your idea is correct, but you've got to be able to verify that it gives you the right answers. So for that reason, sequencing was developed first. And I thought, well, with this restriction enzyme, I could make small pieces of DNA, and I could get into DNA sequencing and go directly at it. And I thought I could probably outdo Fred Sanger because he was still focusing on RNA. So I went down to Cold Spring Harbor and made, I think there were 3 or 4 enzymes were known at the time. And I made them, and they all cut SV40 differently. And we started looking for more. And everywhere we looked, we found them. And by the time the first 100 restriction enzymes were known, 70 of them had been discovered in my lab at Cold Spring Harbor. And that was really nice. It meant that all the postdocs coming in to work with me always had a guaranteed publication because they could publish these enzymes. But it also meant that people discovered that we had this collection. And they used to come to Cold Spring Harbor for meetings with a tube of DNA in their pocket. They'd sneak out of the meeting, come down to my lab and say, do you by chance have a restriction enzyme that would cut this DNA? And so I became very popular at that time. And so we set it up so that it was easy for people to do that. And it soon became obvious that there was a commercial opportunity there. People wanted these things, you couldn't buy them. No one was selling them. I was giving them away. I was sending out strains and protocols and telling people how to make them. And so I go to Jim Watson and I say, hey, there is a business here. Why don't we set up a company, sell restriction enzymes, and we'll use the profit to support the research at Cold Spring Harbor. And he looked at me and he said, oh, that's a stupid idea, he said. No one's going to do that. You can't make money doing this. And plus, it's dirty to be associated with industry. So anyway, that was it. He wasn't interested. And so I looked around and I talked to someone down at NIH who said, well, they'd heard there was a distributor of lab reagents called Miles Laboratory, and they were thinking of selling a restriction enzyme. And I called them up and they said, well, it's not actually us. It's a guy called Don Combe in Beverly. And he started a company and he's going to make the enzymes and sell it to us. And so he said, why don't you talk to him? So I did. Go up, talk to him. And the bottom line of all of that is that I convinced him, A, not to sell the enzymes through a distributor, to sell them directly. I told him which enzymes he should buy, because he should sell rather, because we had this vast experience in my lab at Cold Spring Harbor. I produced a new catalog for him. I gave him a list of mailing addresses. And the bottom line, and he hired me as chief consultant, and the bottom line was they made $200,000 in the first year. This is 1975. It's a lot of money. And they've been successful ever since. I stayed as their chief consultant until 1992, and then moved up there permanently, and regretted not having done it many years earlier. It's a wonderful place to work and to do research. And Don had exactly the same idea that I did, and that was that he wanted to use the profits from the company to support research. And so we have quite a large group now, more than 100 people, doing basic research at biolabs, not with any idea that they have to produce profits or products rather, or anything, but just to do basic research, all supported by the company. And it's great, it's wonderful. Now, one of the things that happened, of course, Jim Watson had hired me to sequence SV40 DNA. And, you know, I basically wasn't doing that. I was making restriction enzymes. And he was constantly threatened to fire me. He said, you know, I don't want you here. I didn't hire you to do this. Why don't you go and find a job somewhere else? But I was lucky. I ran into this gentleman, Norton Zinder, who was a geneticist at Rockefeller, and is probably Jim Watson's only really good friend during his entire career, but he also became my friend. And when he heard that Jim was going to fire me, he would talk to Jim and persuade him not to. So this became a recurring theme. I was 20 years at Cold Spring Harbor, and I've lost count of how many times Jim was going to fire me, but Norton saved me. So always good to have a friend. Now, one of the things we did, in addition to looking for restriction enzymes, the postdocs who came wanted to have some project that could be useful, could be interesting, and would lead to some sort of breakthrough. And this gentleman, Richard Gelinas, had been a student who I met when I was a postdoc at Harvard. He'd worked in Fotis Kafaros' lab, and he and I had kept in contact, and he said yes, he'd come down to Cold Spring Harbor, and in 1974 he became my postdoc and started working with adenovirus, messenger RNA. And we had a question. The question was, at the time, no one knew the sequence, the DNA sequence of a promoter in a eukaryote. We knew the sequence of a couple of promoters in bacteria, thanks to Wally Gilbert, who did the LAC promoter, Mark Potashny, who'd done the lambda promoter. And they were pretty similar, and we thought, well, you know, are eukaryotes the same? And the expectation was that they would be, but, you know, this is something you have to show it experimentally. You can't just guess. And so this was the thing. And we chose to work on adenovirus. We started off looking at the early messenger RNAs in adenovirus, but we could never get enough RNA. Adeno doesn't make much RNA of its own when it first infects a cell, and it's only after it's been going for some time and it's turned off all the host messenger RNA synthesis that you can get lots. So the next thing we did was to decide to look at the late transcripts. And these are the late transcripts, most of them up here. You'll see they all start at different places. They've been mapped quite successfully in another lab at Cold Spring Harbor, so we knew where they all were. And we thought, well, we just need to find, you know, the start point of one of them. And we would do this by getting the sequence of the RNA at the 5' end, and then sequence the DNA that was underneath it. And as soon as you found out where the start of the RNA was coded, upstream would be the promoter. I came up with an absolutely brilliant idea of how we were going to accomplish this. During our work, it was discovered that eukaryotic messenger RNAs have caps. And this cap structure actually means that for any eukaryotic RNA, it has two 3' ends. One at the cap and the other one where the poly A is. The idea then was simple. You take the messenger RNA, cut with ribonuclease T1, which cuts after every G residue, and you end up with a whole bunch of different oligonucleotides. All the ones at the 3' end have poly A on them. All of the ones from the inside, a GP, just end in a GP, and the cap at the 5' end would allow you to do it. And I found a clever way of just capturing these capped oligonucleotides. So the idea was, do the capture, and then spread them out on a 2D gel. And if you remember from the slide, there may be 15, 20 different later RNAs, and we were anticipating there'd be quite a lot of different 5' ends. Well, this was the result. This was early-ish in infection, up to 24 hours, and there are 2 spots, A and B. And after late infection, 15-20, where the only RNA being made is adenovirus, there was just 1 spot. So it meant that all of these 20-odd messages all had the same 5' end. They had the same exact 5' end. And so when you start to think about how that might be possible, well, it could just be a repeated sequence. Turned out not to be true. We got a bunch of them, a bunch of different messenger RNAs, and there were no common sequences at the 5' ends. And so the question was, what's going on? Well, Jelinas and I wrote a paper showing that all of these RNAs, there were actually 11 nucleotides in this cap structure. All of the later RNAs had exactly the same cap structure. So the question was, how is that possible? Well, could have been a repeated sequence, but we'd already shown that was not true. But then we had a very puzzling result. We thought we will pull out RNAs from different parts of the adenovirus genome, which you do by hybridizing the RNA to DNA and then purifying it. It turned out if that RNA hybridized to the DNA was very gently treated with ribonuclease, you lost the cap structure. It disappeared, just like the poly-A structure, because neither was coded right where the main messenger RNA was. So then we came up with an idea. We thought, well, maybe there's a primer. Maybe the primer goes and binds at all these different start sites, and it makes the messenger RNA, and they all have this primer at the end. And that seemed attractive because there was an RNA encoded by adenovirus called VA-RNA. We knew where it was coded, and we thought, well, maybe that is the primer. And so we devised an experiment broadly shown here. So we thought the RNA structure was such that it matched the DNA, but at the 3' end was poly-A, which would stick out, and at the 5' end, the primer would stick out. Turns out if you take DNA and heat denature and add messenger RNA, you get R loops formed. And these R loops formed, and they're stable because DNA-RNA hybrids are more stable than DNA-DNA hybrids. And so we thought, well, if we could guess where this 5' end was being located, we could make a DNA probe, just like this, and find the 5' end and show it look like this. And you could then look in the electron microscope, you could see it, and so on. So great idea. We had the idea on a Saturday morning. Unfortunately, neither Richard nor I were electron microscopists, but we had these 2 very good electron microscopists who worked in the lab down 1 floor below us. And so we went down and we said, you know, if we make the reagents, could you do this experiment for us? And they looked and they said, well, it's never really been done, but yeah, it looks okay. It looks as though it should be doable. So we set out, made the reagents, and on the Tuesday morning, so the idea came on a Saturday, Tuesday morning, take the reagents down. Louise Chow, the lady here, she was fantastic electron microscopist. The very first molecule she saw looked exactly like that, well, almost exactly like this. Instead of hybridizing at just 1 point at the 5' end, it actually hybridized at 2 points. And so what had happened here was that this piece of DNA, the probe DNA, contained the genetic information for the 5' end, but in a little bit here and a little bit there. And this was the discovery of splicing. The more she looked, all the molecules looked like this. Everybody was really enthusiastic. I have to tell you, before then, everybody had said we were wasting our time. They said we're looking at an artifact, you know, we're just not doing the experiments properly. But Richard and I were pretty confident we were. And this really proved the point. We had all the biochemistry evidence that was necessary to support this. You know, very often when you see something for the first time in the electron microscope, it's just one molecule, and so you start to worry a little bit about it. But we had all the biochemistry to support it already, and so by that Tuesday afternoon, people were already talking about Nobel Prizes. We were telling everybody about it. Many people went and looked at their own data, discovered that they could explain data that they'd previously not been able to explain in terms of RNA splicing and the genes were split up into pieces. So that was very exciting. This is Tom Broker, Tom and Louisa, man and wife, and they were the two electron microscopists. So we published this paper. Title was An Amazing Sequence Arrangement. I had a hard time justifying the word amazing to my co-authors. They said, you know, it's not scientific. You really can't put that on the title of a paper. But eventually they gave in. We sent it out for review. It went to Cell. There were 4 reviewers. Came back, every one of them said the same thing, that this was a great paper, wonderful discovery, but you can't use the word amazing in the title of a paper. It's not scientific, you know, it's not right. And so I got on the phone to Ben Lewin, who was the editor of Cell, and I said to him, you know, we were talking about some of the points that the reviewers had raised, some of the minor points. And during the conversation, I said, hey, Ben, when you first read about this, what did you think? He said, oh, it was amazing. I said, I rest my case. And that is how amazing, as far as I'm aware, this is the first time it was ever used as the title of a scientific paper. Now, in 1993, Phil Sharp, who had made a similar discovery totally independently, he was not related in any way, although he and I were at Cold Spring Harbor when I first went there, and we have one paper together on mapping adenovirus messenger RNA, but he made the same discovery independently, and so we shared the prize in 1993, and I have to tell you, it is just wonderful to share the prize, to get a Nobel Prize. I learned about it from CNN. I was up early in the morning, 6 o'clock in the morning, writing a paper, and the phone suddenly rang, and we have a phone right next to our bed, and my wife was asleep, and I'm thinking, oh shit, you know, I really don't want to have to answer the phone, so I did go quickly, because I was afraid, you know, she would wake up and I wouldn't get any more work done. And the guy at the other end said, this is CNN calling, have you seen the news? I said, why the hell would I be watching the news at 6 o'clock in the morning? And the guy said, well, it's just been announced that you've won the Nobel Prize. So it didn't seem quite so bad after that. So that was nice. So anyway, we got all the preparations together, went over to Stockholm. Just wonderful time in Stockholm. Very good. Now, one of the things you do when you go and pick up the prize is you have to give a lecture. And almost everybody who gives that lecture talks about all the wonderful work they've done following up on the discovery. Now, after we made the discovery, a lot of the work that had to be done was electron microscopy, which was not us. I tried to set up an in vitro splicing system, and we picked the wrong RNA polymerase and couldn't make nice messages, and Tom Maniardis picked the right RNA polymerase and got that going. And so we ended up that I just stopped working on this for a few years, and we switched over to something else. We started working on DNA methylation. So when it came time to give my Nobel talk, I talked about a discovery that we just made a couple of months before I had to go to Stockholm. And that was that when a DNA methylase is going to methylate one of the bases in DNA, it flips that base out 180 degrees right into the active site pocket in the enzyme, puts the methyl group on, and then it flips back. So this was the first discovery of base flipping. And so I thought, you know, this is nice to talk to the Nobel Committee about that. You know, maybe they'll think it's a good thing. No further follow-up yet, but you never know. Anyway, it turns out base flipping is quite common. There are a lot of enzymes that do chemistry on DNA. Anyone that does chemistry on the base usually uses base flipping as a way of getting to the base and doing its thing. Transcription also uses base flipping, and DNA replication, we think, in some cases does too. So it was a nice discovery. That discovery was made with Xiaodong Cheng, who was a postdoc at Cold Spring Harbor at the time, and we'd been working on trying to get a good crystal structure for 2 or 3 years, and we just got this just before I had to go to Stockholm. I want to take a little digression here and tell you a little bit more about New England Biolabs. As I told you, Don Combe, who was the founder, had started this company in Boston. The company at the time was Don, his wife, and one technician. They were in a basement in Beverly underneath a hairdressing salon. They had a makeshift lab, and they bought used equipment from Harvard. Don himself had been an associate professor at Harvard Medical School, and they didn't give him tenure, and he got very annoyed about this, and he said, well, you know, I'm going to leave the academic life and start a company, and I'm going to use the profits to support research, which is what he's done. So in 1992, I moved up there as research director, and we now moved to this new building in 2005. The whole company is now about 650 people, of whom 350 or so work in Ipswich, and the rest are around the world, mainly selling in distributing and so on. This was the very first catalog that I put together, and I'm sure all of you have seen company catalogs since then, and they always have a whole bunch of stuff in it that is not just product oriented. This was the very first catalog to do that, and we had a list of the known restriction enzymes at the end and a little bit about them. This was BioLabs between 1980 and 2005. This was this building in Beverly. Then we moved out to Ipswich, put up this brand new building on a big site out there. It's actually interesting, the site used to belong to the Roman Catholic Church, and we had to get permission from the Pope in order to buy it. Apparently, you know, because it belonged to the church, they are very concerned about who buys it. And so I always thought the next catalog we should put a picture of the Pope waving out of his Pope-mobile, but our marketing department didn't think that was a good idea. So anyway, but we had extensive lab space. We ended up an aquatic water purification system, solar. It's probably one of the cleanest labs out there. So Don started off as the CEO. Our current CEO is Sal Rosella, who took over about 1.5 years ago. I'm Chief Scientific Officer. 75% of the people who work there, no matter what department they're in, have scientific training. So we're very much a company of scientists. A very cooperative environment. I'll give you an example. During COVID, we were making the enzymes that Moderna needed to make their messenger RNA. So there were 3 enzymes, 3 methyltransferases to modify the cap structure, and they bought them all from us. And this meant we had to scale up production almost 10,000-fold to produce enough to satisfy the world's needs for Moderna vaccine. We also made RNA polymerase, and that was sold to Pfizer, and they bought that for their vaccine. So we played a major part in vaccine production. But we had to scale up enormously to do that. And the company is such that if you ever ask for a volunteer, typically you get 20, 30 people will say, oh, I'll do it, I'll do it, I'll do it. And so we didn't have trouble scaling up. The people just were anxious to help and do what they could. And it's very typical of the company that we have a wonderful atmosphere within the staff, and we're more like a family than we are a company, to be honest. One thing, you know, I do in my life, I like puzzles and games. I learned to play Go. Many of you, if you saw the slide of my time at Harvard, Kazuo Kurosawa taught me how to play Go. We used to play Go by mail after he left for quite a long time. Also taught me how to play shogi, which is Japanese chess. And I love chess. I was the West of England junior chess champion until I got beaten by this little kid. I was defending my title, this little kid sitting on a chair, on a cushion on a chair across from me, and I start playing with him and he thrashed me. It was, terrible. But, you know, that's the way it is. These young people, they're going to take over the world one way or another. I think we can't stop them. So anyway, I love chess. I also love croquet. So this game, croquet, I learned to play that when I was on sabbatical in Sidney Brenner's lab in Cambridge back in 1978. And I went down to King's College. They have wonderful lawns down there. And so we went down for the lawn, and there were a number of people who were busy playing croquet down there, and we had a foursome, and I learned to play. I loved it, fell in love with the game. So when I won the Nobel Prize, I spent a good portion of my Nobel Prize money put in a croquet lawn in front of my house. And there was a little article in the Scientific American about this, in which the authors were looking at how people had spent their Nobel money. And they were saying, you know, oh, this person gave it away to this, and this person did this and that and the other. And then one of them, Richard Roberts, put a croquet lawn in, frittered away his money on a croquet lawn in front of his house. But I've gotten more enjoyment out of that than I'm sure all these people who gave their money away, and I couldn't have afforded it otherwise. So it was good. That was a good thing. This was something else, indication. This is actually taken on my croquet lawn. I was the stud muffin in the Stud Muffin of Science calendar. I was Dr. December in 1997. And I don't know if any of you know about the Ig Nobel Prize ceremonies. They have them every year. They're in Cambridge. And one of the people who helps to organize that also organize the Stud Muffins of Science, and I go and participate in the Ig Nobel ceremonies every year. They're a lot of fun, and I like fun, I like jokes, I like to laugh. Laughter is so important. You know, I don't care what's going on, if you see a good joke, you can have a laugh and you'll feel much better for it. I also got a few honors. I was knighted by Prince Charles at the time, now King Charles. You know, the worst thing about that, if you look at the sword, it's got a very sharp edge on it, and so, you know, you always worry when he's pressing down that he's going to take a slice out of you. But he didn't. I survived. It was okay. But that was kind of nice. It really is very nice to do all of that sort of stuff. I got an honorary degree from the University of Bath, where I'd grown up, and also they made me a Freemason or something of the city. Anyway, they gave me the keys to the city. The school that I went to put in a new science building, and they named that after me. And then the University of Sheffield also. I was their first graduate student ever to win a Nobel Prize. The first student ever to win a Nobel Prize there. And so they were very happy about that. And they put up this building, named it after me. But 3 years after I got the prize, another chemistry student, Harry Kroto, got the chemistry prize. And so he was 3 years ahead of me. And so we often used to appear on stage and talks about things, and he would always say, oh, you know, I taught Rich everything he knows. You know, we never actually met when I was at Sheffield, but just because he was older. And so I would always respond and say, but Harry, how come it took you so long to get your prize? So anyway, we used to have a lot of fun with that. One of the things that I discovered after I won the Nobel Prize is that, you know, I would start to say things and express opinions about things and complain about things or suggest things that people might do. Before I won the prize, you know, occasionally people would listen. They never actually did anything. After I won the prize, everybody listened. And not only that, a lot of people acted upon it. They would then follow through and do things. And so I thought, well, you know, this is rather good. Maybe one could use the Nobel cadre, if you like, to do some good. And I started with an open access movement that the Vice Chancellor mentioned, because I've always been very keen on open access to scientific results. I think we had 40 Nobel laureates that I rounded up. We wrote a letter to Nature. And it made quite a big difference. I was at the time chief editor of Nucleic Acids Research, and they were one of the first journals to go from a typical model where the journals are trying to make money to an open access model. So that was good. And I've done a lot of other campaigns. One, the most successful, there was a case of some nurses and a student in Libya. And they had been charged with spreading HIV among the children in the children's hospital in Benghazi. There were 400 children that got HIV. And Gaddafi decided, first of all, that it must be Israel that had caused it, Mossad, but then they quickly got over that. And then they decided these nurses must have done it deliberately. They arrested them, tortured them, several of them confessed in order to stop the torture, and they all ended up on death row. And when I first heard about this, I couldn't believe that it would go as far as it did. But then a reporter from Nature wrote to me and said, are you aware of what's gone on in Libya? And because they were on death row, they were going to be executed. And so I mustered the Nobel laureates. I think we had about a little over 100. And we wrote a letter, and I took it down to the Libyan ambassador to the UN, and I said, hey, this is wrong. The scientific evidence just doesn't support the fact that they did it. What the problem is, they share needles. So, you put a needle into an infected person, you just spread it from one person to another, and that was what had happened. So I go to the Libyan embassy, and I say, would you give this letter to President Gaddafi? And the ambassador was very nice, and he said, yes, I will do this, and sent it on. And then I never heard anything, and after 2 weeks, I got a phone call from Gaddafi's son, who said, would you come to Tripoli and talk to us about this? So I got on a plane, I go over to Tripoli, met with him, we met late one evening, spent about an hour with him, and at the end of that time, he says, we're going to let them go. And so we were able to get them off death row, out of Libya. But it wasn't just because of what I personally did, but the fact that when you get a lot of Nobel laureates all saying the same thing, a lot of organizations listen, people who can do things. I'd contacted the Foreign Office in London, and they'd been very active. They'd talked to other European ambassadors and so on. So it all worked out well. But I've discovered that Nobel laureates can do a lot of good when we unite and perhaps get other people who are in a position to do something personally to actually go ahead and do it. Now, I want to talk to you about the importance of luck. I've already told you about billiards and snooker and how this old gentleman said, you have to take advantage of luck. Kazuo Kurosawa, I was put in a lab. He was a postdoc there. I could have been in any lab, but it was him. And he made a huge difference. I think I learned more about doing experiments in chemistry from him than from anybody ever. Ended up at Harvard instead of Wisconsin. Now, Wisconsin was a good school, but Harvard was better in the sense that there were so many more well-known people there, and you have the opportunity to interact with loads and loads of really good people. Looking for eukaryotic promoters in adenovirus. This is a good example of how failure can be such a good thing. I came up with this great experiment. It was everybody agreed, wonderful experiment, but it didn't work. And so what we were trying to do failed miserably, but we did a post-mortem. We tried to figure out why it had failed. And of course, it failed because nature was trying to tell us something. Nature was trying to tell us that what we thought about the way transcription worked in adenovirus was very different from the way that it worked in bacteria. And so failure is really good. When you do an experiment and it fails, don't give up. Repeat it. Make sure that it wasn't because you spat in the tube or something, but make sure that you did the experiment properly. And if it continues to fail, do a post-mortem and work out why. And maybe you too will discover that nature is trying to tell you something that would be very helpful. The fifth one I haven't told you about yet, and this is probably one of the luckiest things that ever happened to me. You remember the plane that went into the World Trade Tower in New York on 9/11? I was booked on that plane until 2 weeks before it took off. And I was going to a meeting in California, and the meeting got moved forward by 1 day, and so I had to change my ticket, and I took that plane the day before 9/11. So I was in California when that happened. That is luck. Now that is real luck. If the meeting hadn't been cancelled, I would have been on that plane. I wouldn't be here talking to you today. So that was very lucky. I also had another interesting break. There was a guy called the Unabomber in the US. He was an assistant professor in mathematics at MIT, and he went off the rails. He was nuts. And he started sending letters and bombs around the country to various people. And the very last batch of letters he sent out 5 letters. One had a bomb. I got one of the letters, but I didn't get the bomb. That was pretty lucky too. And then shortly after that, they found him. His brother turned him in, and they arrested him, and everything was okay. And then finally, all of this started at Cold Spring Harbor by studying restriction and modification systems. And I still study these systems. I still work on DNA methylation. And I'm really happy because it turns out that the DNA methylases that protect bacteria against restriction enzymes do other things too. We made a discovery just a few years ago about an enzyme, a DNA methylase in Clostridium difficile, that controls sporulation. If you knock out that methylase, Clostridium difficile cannot sporulate anymore. And if any of you have ever been in hospital, you will know that it's very easy when you're in hospital to get a C. diff infection. It's a very nasty organism. And the problem is that there's no good cure for it because it forms spores. So, you treat it with antibiotics, it forms spores, and then just sits there until the antibiotic goes away, and then it starts growing again. And so if you could stop it from sporulating, this may make all the difference. This could be really good. And so these methylases have a lot of interesting roles other than just serving as protection against restriction enzymes. And I heard a very interesting theory some time ago. I was giving a lecture, a remote lecture, to an audience in Novosibirsk in Russia. And one of the people in the audience says, is it possible that the restriction enzyme there is there to keep the methylase honest, to make sure that it doesn't change its specificity, because it's actually the methylation that is important, much more so than the restriction enzyme, and the fact that these systems protect against phage is just by chance. And I thought that was fascinating, and I'm continuing to work on that. I've been trying to find out who came up with this idea, and I've been totally unsuccessful. So if by chance any one of you in the audience came up with the idea, please tell me. I would love to give credit to this individual. And I just want to finish by telling you about 3 books and a film that I've come across recently. The first one is this film here. It's called Picture of a Scientist. It's about a lady called Nancy Hopkins and about other scientists, lady scientists, who've been discriminated against by the academic system. It's a documentary. It's very well done. There's a book called The Exceptions by Kate Zanicki, and this talks in more depth about Nancy Hopkins and the way she was at MIT and realized that she was being mistreated because she was a woman. And she went and documented all of the ways in which she was being mistreated, goes to the president of MIT, and they changed the rules to accommodate the fact that they were discriminating against women. Another very good book is by Kati Carrico. She won the Nobel Prize this last year for the discovery of messenger RNA and how to make it and the fact that it could be used as a vaccine. Her story is in many ways tragic, but it's just wonderful that she survived everything that happened to her. It's autobiographical, it's great, really a good book. And finally, this book over here, Invisible Women, is written by an English author who documents and has huge amounts of data about how women get mistreated by men. And I think every man who is in science should read this book, because we end up mistreating women without realizing it. And one of the very common things we do is we treat them as though they were men, not recognizing the fact Women can have children, men can't. Women have periods, men don't. If you look at the medical history, almost all medical experiments for a long time were only done on men, not done on women. And I've been really impressed by this, because after I read it, I looked back and I realized I'd been doing this stuff and just hadn't realized it. And I think it would be good for everybody to read these books. They're really good. And so I'm indebted to all of you for listening. I think we're all indebted to the ladies in our lives, and especially the lady scientists, so that's why I wanted to end up with this. So I thank you all very much. Yeah, sure. Okay, thanks.
Interesting and tough questions. I was wondering whether you are contemplating of writing a book on the importance of luck yourself.
Well, I'm writing a memoir, so in it there is a great deal about luck and many things. You know, I've been lucky in many other ways. I don't have time to talk about everything, but luck is really important. I was just at a meeting in Dubai, and there were 5 other laureates there, and we were on a panel, and every single one on that panel said, well, luck was the reason that they made their Nobel-winning discovery. They were doing something else, didn't work, and luckily was a big discovery. And I think it's important to note that, you know, never ever make it your life's goal to win a Nobel Prize, because you'll be disappointed. It's unless you happen to have that stroke of luck that makes and leads to a big discovery. And we all have luck, and sometimes the discoveries are small, sometimes they're big. If they're big enough, then maybe you can get a Nobel Prize, but you can't aim for this. All you can do is do the best science you can. Do that and you'll have a very happy life.
Thank you. Microphone. Or you can use this. Take this.
Yeah, I can say.
Say it anyway, you can hear.
Once the Nobel Laureate can influence the leaders and opinion, there are so many wars are going on, Ukraine war and Israel war. And whether Nobel Laureates can come together and write a letter to these leaders, and whether you can do something about this.
Yeah, well, we do that. We actually wrote a big letter about the Ukraine war. We had 203 Nobel laureates. There are only 300 alive. We had 203 laureates signed up and told Putin and co that they better stop. But didn't have much effect yet. So.
So I was expecting a different lecture, but I'm glad we had this lecture. And probably in a good way. I wanted to ask a few technical questions. The first one is there a way to identify specific splicing patterns for different diseases, which could be a way to kind of manage those? And to add to that, are there models to predict alternative splicing patterns or events which could kind of be specific to certain conditions, as well as for understanding the evolutionary relationship with the RNA processing pathways?
Yeah, so we, shortly after we made the discovery, one of the things we wanted to do was to see if there were common signals that we could recognize bioinformatically. Bottom line is, you can't, even today, you can't do a really good job other than by sequencing the messenger RNA, sequencing the DNA, and seeing where the junctions are. In terms of the clinical importance of this, alternative splicing turns out to be quite important And a postdoc of mine, Adrian Craner, made a huge discovery a few years ago about spinal muscular atrophy, where he discovered that it was caused by a defect in alternative splicing, and he found a way to correct it. And so there are now children who used to die by age 3 or 4 who are living 10, 12 years already because of this breakthrough. And we know that there are many diseases also that are caused by aberrant splicing, problems with alternative splicing, and similar methods should be available for those too.
Thank you, sir. Just supplementary to this, do you think something like alternative splicing or nature actually believes in luck?
I'm sure it came around by luck, but it was selected for after it happened, right?
Thank you, sir.
Yeah. Here.
You talked about your love for mathematics in the early days, but can you tell us a little bit about the importance of mathematics and now AI in biology?
Sure. So, what I do today is all bioinformatics. So I'm looking at methylation in bacteria. I work on bacteria. And we're really interested in what the methylation is doing. And so I design computer programs, and mathematics has been constant for me. I first got involved in bioinformatics back in 1978, before that, 75. Before there was even such a thing as bioinformatics. The very first programs to assemble DNA sequences we wrote, for instance. So a lot of stuff of the early programs we did then. I was telling somebody the other day, I used to love programming. And one day, I just debugged the program. I looked at the clock. It was 8 o'clock in the morning. I'd worked all night. I hadn't even realized I'd worked all night. And I had to go home and explain to my wife that I'd been programming, not doing something else that she might not have approved of. And then a few days later, exactly the same thing happened, and I quit programming, and I hired a programmer at that time. So, bioinformatics is incredibly important. And certainly machine learning, we got involved in a lot of neural network programs early on. AI can really do wonders, but AI is only good if you have really good data sets. And so part of the problem with AI these days is that a lot of the generational AI is just using very imperfect data sets. It's going on what it can pick up from Google or books or whatever. And the database is not that good, and so it will make mistakes. But in science, we really should be able to produce a very good database. You can't just take the literature. You've got to prune out all the rubbish that's in the literature so that you end up with just those good articles. But fortunately, there's stacks and stacks of good articles. And so I've actually been talking with the leader of the National Academy of Sciences in the US about perhaps trying to get some of the Academy members to help put together a really reliable database of information that scientists could use to try to do better. There's no doubt it will be good. It will be very valuable going forward.
I have two quick questions. The first one is, you have emphasized on luck. So what do you think, luck or serendipity, which is more important for scientific discovery?
It's just another name for the same thing. Serendipity, luck, it's the same thing.
Well, there could be...
They're synonyms.
Yeah.
Right?
The second question is that,
luck.
almost in the similar line, the luck favors the prepared mind. So what do you think on the preparation of mind for lucky discoveries?
Yeah, well, luck, I think that the origin of that statement is that people who were lucky like to convince themselves that they did something special ahead of time. I don't believe it. I really don't believe it. I think you can take advantage of luck when it comes your way, but you can't prepare for it, you know. Luck is luck. You never know. I mean, you know, here I am, I'm 2 years old, my side of the street.
Literature to promote equality and diversity in science, because surely that is good for science, right?
Oh, I couldn't agree more. So one of the things, I've been talking to some of my Nobel friends, and one of the things we've noticed is that many conferences like to get Nobel laureates to come and speak, and they're almost all male, because the Nobel Committee has been very biased in favor of males. And so quite a number of us now have said, well, whenever we see we're invited to a conference, we want to know, have they been inviting female speakers to come along so that we have equal numbers? Because there are some wonderful women, wonderful speakers. And when it comes to students, a female student is much more likely to be inspired by a female who's done well than by a male who's done well. You know, I don't know too much about the hurdles that you face, and I can't talk about that, but women can. So that's one thing that I'm trying to get more Nobel laureates to do. And I'm also personally pushing at the moment. The head of the Nobel Foundation is just resigning, and there's a lady who, with any luck at all, will take his place, and that I think could have a huge effect. She's terrific. She's really good.
I want to ask one more question.
Okay.
Since you are very candid, that's why I'm
Too
asking.
candid, perhaps?
Yeah, in this business of winning Nobel Prizes, we hear there is a fair amount of politics, fair amount of discrimination.
Discrimination for sure. I'm not sure about politics, but one of the things is you have to be nominated before you win a Nobel Prize. Phil Sharp, beginning in 1978, was nominated for all sorts of prizes. He won, I think, pretty much every prize going before he won the Nobel Prize. I won nothing. The Nobel Prize was my first prize, and I think this was because Jim Watson and I fought all the time, and he never nominated me for anything. So you have to be nominated, right? And some people, well, some people go out and they lobby for these prizes. I know, because they write to me and say, will I nominate them? So I'm not a lobbyist. I don't believe in lobbying.
So one question I have here is that, in terms of molecular biology applications, we talk about gene therapy is one of the things which is coming on.
Yeah.
So we have been talking about gene therapy from last almost 15-20 years. So according to you, how far we are up to like treating the diseases which are coming because of the mutations happening in the individual, and like we are treating or modifying the bacterias, like methylation, restriction in genes. Is there is a way that we can treat mutations happening in individuals by medicines made through the gene technology?
So those are really two separate questions. So in terms of doing gene therapy, you're probably aware of the recent work that's gone on in England where they can solve sickle cell disease by turning off or turning on the primitive hemoglobin production. So that's that's the first positive example. It's been approved as a clinical medicine for sickle cell disease. There are many others in the pipeline, but they still have a ways to go. But the microbes, which is actually something I'm much more interested in than all this other stuff, the microbes are absolutely key to our lives. If I kill all your bugs that live with you, you will die. You can't live without them. They keep us alive, they take care of pathogens, they do so many things, and we're only just starting to learn what they're capable of. You know, we know there are bugs that don't like us to get cancer, and so they will kill cancer cells. Very interesting. One of the problems, though, is if you've got a bacterium that you think is going to be really good for you, we don't know how to get it established in the gut or elsewhere on our body yet. We will learn, but we don't know how to do that at the moment. But I think there's going to be some wonderful opportunities going forward. The microbiome is fantastic, beautiful, lovely. I love bacteria.
Do you think in future we may eat the microbiome as a drug to treat our gut biome?
You may well do. You may
Thank
well
you.
do.
There is one question there. Two. Do we have time limit here? You're okay? One or two?
Good afternoon, sir. It was a very delightful and inspiring talk. So I want to know, according to your perspective, that in science, what matters more, the question or how you answer the question?
Well, you know, I think both matter, and I wouldn't say that one was more important than the other. Asking good questions is always good. It's always good. And you know, if you look at young children, they typically ask wonderful questions, and then we send them to school and we knock all of this curiosity and creativity out of them. So I think we could do something better there for educating the children. But the way you go about trying to answer a question is also important. You know, we were looking, in our case, for a promoter, and we went about it in what looked to be a really good way to do it, but it didn't work. And so I think also when you're trying to answer questions, you also want to make sure that when things fail, you do a post-mortem to find out why it failed. You know, one of the reasons that flying in an aircraft is so safe is because every time there's a crash, they do the most incredible post-mortems to find out what went wrong. And that has made actually flying one of the safest things we do. It's much safer than walking across the road. Many more people get killed from walking across the road than die in plane accidents.
Thank you, sir.
Yeah, I think on that note, there is a separate session arranged for question answers. And Dr. Roberts will answer many questions later. So hold on to your questions and attack him with questions.
Okay.
Thank you.
Okay. You're welcome.
I would request Professor Anand Kumar Kondapi and Professor JSS Prakash to join our Vice Chancellor in felicitating Sir Professor Richard Roberts.
Thank you. Thank you. Thank you all. So, have a little cup. Which sort of the caption, and if you keep the cup on your table, you will remember. Okay. Okay. Thank you. You know about this pen cup. Thank you, everybody.
So we will break for tea now, then we will assemble again for the next round of session.
Okay.